Today on In Your Face Anatomy, we're doing cellular transport. Now get out of my face.
All right, cellular transport. So last time we talked about all of the components of the cell, including the cell membrane and its phospholipid bilayer, how it forms those two layers of phospholipids to separate the contents and help protect our cell. So now we're going to talk about how we get things in and out of the cell, getting through, around, over, bypass, whatever, past that membrane. So with cellular transport, we really have two types of processes. I have passive processes and I have active processes that we need to look at. So my passive processes get their name because they require no ATP. We don't have to spend any energy to make this stuff happen. So I got simple diffusion, facilitated diffusion, and osmosis. With my active transport, I do have to spend energy. This is something I have to make an investment with in ATP to make this stuff happen. So we've got active transport, endocytosis, exocytosis, and transcytosis. So with passive transport, I want you to know no energy and be able to tell me which processes are passive. That is simple diffusion, facilitated diffusion, and osmosis. Active transport does require energy, and I want you to be able to tell me active transport, endocytosis, exocytosis, and transcytosis. So that's the difference between passive and active. Now, before we get into discussing these individually, I want us to understand the concentration gradient. The concentration gradient is literally just going from high concentrations to low concentrations. That's the natural order of things. Particles tend to travel in a straight line until they hit something and bounce off. And if there's not a whole lot of stuff in front of them, they're going to continue to move in that direction. So that's why we go from high concentrations to low concentrations in the natural order of things. The concentration gradient, high to low. It's just the way to do it. So now that we have that established, let's look at our first process, simple diffusion. So simple diffusion is really easy. It's just spreading things out evenly. I tell people simple diffusion is like farting in a room until it spreads all the way out. You know, it's really bad if you're close to the fart, but eventually it spreads out all through the room, and it's hard to notice at that point unless it's a bad one. You know who you are. So it's just spreading things out evenly. Doesn't require any energy. It's one of our passive processes. When everything does get spread out evenly throughout the solution, then we've reached diffusional equilibrium. So that is when substances become uniform. When substances become uniform, then we've reached diffusional equilibrium, and that's really as far as diffusion is going to go. Now, we do have a condition known as the physiological steady state in our body. Physiological steady state, this is when concentrations of diffusing substances are unequal but stable, just meaning we never reach diffusional equilibrium with the physiological steady state. So we use oxygen as a example for the physiological steady state because all of our cells in our body are constantly using up their oxygen. We're using them to break down ATP primarily, and we're delivering oxygen to all cells of our body. So we never have as much oxygen in as we do out because we use it as soon as we get it. And so the physiological steady state defined is when concentrations are unequal but stable. So by us constantly using oxygen, that always we have a pull for always getting more and more oxygen into the cell. That is the physiological steady state. So make sure we can make the distinction between these two. Diffusional equilibrium, when everything is spread out evenly, we're good. The physiological steady state, concentrations are unequal but stable. So when it's time for diffusion to occur, I've got us a little cell membrane drawn over here. I've got some phospholipids. Remember, we have that phospholipid bilayer. We talked about phospholipids earlier. So I've got my phospholipid bilayer set up and my phosphate heads that love water. So they're pointing out into my extracellular matrix. This is my cytoplasm on the inside here. And where their fatty acid tails come together, I've drawn it in yellow here. We get this fatty lipidy layer. And that's what stops anything that is water-based from getting into the cell. So if I was to drop some particles out here, boom. And we wanted this to diffuse. I'm going to go put just a couple out here. I don't need a whole lot to show this. There we go. So we got a couple particles out there, and we want diffusion to occur. This only works if whatever we are trying to get to diffuse is lipid-based, because we have this yellow lipidy fatty layer that's going to stop anything that's water-soluble from being able to get inside of our cell. So this only works if it's lipid-based for simple diffusion across the cell membrane, I should say. So it so happens, we'll say these are some steroids. They are lipid-based. So we're going to spread those out for diffusion to occur. Leave a couple on the outside out here, spread out pretty evenly, just for diffusional equilibrium references. Boom, there. Now everything is spread out equally. This is diffusional equilibrium. Simple diffusion, we're just spreading it all out. Spreading it out. That's all we're doing. All right. Our next type of cellular transport was facilitated diffusion. Oh, don't fall. There we go. Okay. Facilitated diffusion. So facilitated diffusion is just like our simple diffusion. It's really the same thing. It works under the same principles. It's just regular diffusion, but now we're using proteins. With simple diffusion, with regular diffusion, we could just go straight through the membrane. So this is for substances that are not lipid-based, substances that can't just pass through the membrane. They have to find an alternate way in, so we use facilitated. So facilitated is just like helper. So this is helper diffusion. We need something to help us to get these particles to diffuse. So we primarily are using various proteins. Proteins are the big thing with facilitated diffusion. It's simple diffusion, but we got to use proteins to make it work. It still follows the concentration gradient, just like simple diffusion does. So we're going from concentrations of higher concentrations to lower concentrations, that natural process. But the number of proteins that my cell has is going to limit the rate. That just means facilitated diffusion can only go as fast as my proteins will allow these particles to pass. So it's a little bit slower than simple diffusion, but it does bypass that lipid bilayer. So if I pull my cell membrane back out here, let's get rid of my lipids that we drew in last time. All right, so this time we're going to do, I don't know, we're just going to say something that's water-based. We'll just say water. Try to get some water into the cell. So I'm going to draw it in green this time. So here's my water. Boom, boom, boom. It can't go here because it's just going to bounce right back out. It's not going to be able to get past that lipid bilayer. It just won't let it through. So this is one of my carrier proteins that we're going to use for facilitated diffusion. What it is, it's like a pathway, almost like a little hallway or a channel. So my water molecules are going to pass into this channel pathway, this carrier protein, and it's going to pop out the other side. And in that way, we're able to bypass that lipid bilayer altogether. But remember, we can only move as fast as we have proteins to help the process along. So that is facilitated diffusion. All right, our next type of passive transport is osmosis. So the first thing I want you to know about osmosis is that we are always talking about the movement of water, moving water. That is osmosis, moving water. You hear water, you should think osmosis automatically. So just like all the other passive processes, it follows the concentration gradient. So we go from higher to lower regions of concentration. Now we have something new with osmosis where we have to understand osmotic pressure. So osmotic pressure is defined as the ability of osmosis to be able to lift or move a volume of water. So there's enough pressure there to lift or move a volume of water so we can equal it out on both sides. That's really what we want, the same amount of water on both sides if we can get there. Now water always moves towards a region of higher osmotic pressure. That is key. Let's put a little star next to that. Water always moves towards a region of higher osmotic pressure. And so I've got in my little picture here, it's kind of hard to see. Let me change that. So I've got a little flask of water here, and I've got it separated right down the middle by this semi-permeable membrane. I've got this little membrane separating it right in the middle. And at first glance, it looks like we've got the same amount of water on both sides, but we don't. What we have are these purple rocks in it. Well, they're solutes, essentially, is what they are. Solutes, just anything that's in the solution. So I've got more of my purple solutes on the left side over here. We can see that I've got 6 of them on the left. I've only got 3 on the right. So these guys, these purple solutes, are taking the room where water molecules could be. So we actually have less water on this side than we do on that side because we have less solutes. So what's going to happen with osmosis is it's going to want, with this semi-permeable membrane separating the two, water's going to move to the opposite side until we end up with the same amount of water on both sides. So we're going to end up with something along this line where it's going to even out. Now we've got the same amount of water on both sides once we take into account the solutes. So something important to understand here is the higher the solutes, the higher whatever it is, the concentration inside, higher the solutes is going to equal the higher osmotic pressure. So we just said that water moves always, bam, 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 always moves towards the region of higher osmotic pressure. But the higher the solutes, also the higher the osmotic pressure. So water will also move towards the region of higher solutes if it's not already reached that equilibrium. So, osmosis. Osmotic pressure, moving things along. So let's apply some osmosis practically. So we have 3 kinds of solutions. Tweak that a little bit. There we go. Okay, 3 kinds of solutions. We have isotonic solutions, we have hypertonic, and we have hypotonic solutions. So iso just means same. So isotonic is the same osmotic pressure as our body fluids. So if we have the same osmotic pressure, which way is water going to go? So if I take a cell and I place that cell in a isotonic solution, what's going to happen? Which way will water flow? Nowhere. Nothing is going to happen. Nothing happens in an isotonic solution because we don't have any pull on the water. Water's going to want to go to the higher osmotic pressure, but there is no higher osmotic pressure. We have the same osmotic pressure, so nothing happens if we put a cell in an isotonic solution. Next up, we have a hypertonic solution. So with a hypertonic solution, we are going to have higher osmotic pressure. That's what hyper means, higher. Think of hyperactive, high energy. Hypertonic is higher osmotic pressure than our body fluids. Higher osmotic pressure. So if the solution here is hypertonic, then that's got the higher osmotic pressure. We put a cell in there. We just said water always moves towards the region of the higher osmotic pressure. Well, that's the solution. It says so right there. As soon as I see hypertonic, I know this is the higher osmotic pressure. So water is going to leave my cell, say H2O out. Water leaves the cell, and that's going to cause our cell to shrivel up as the water starts to shrink away from it. Boom. It's going to shrivel up or shrink. So hypertonic, the cell shrinks. If we put it in a hypertonic solution, I'm just going to get rid of the whole thing. Bam. And lastly, we have a hypotonic. So hypo means low. Hypo means low. So what happens if we put a cell in a hypotonic solution? Well, if this is the lower osmotic pressure now, that means the solution inside of my cell is hypertonic. If this is hypotonic, this one's got to be hypertonic. So that means water is going to move from the solution into my cell. So water in. Water flows into my cell, and that's going to cause my cell to swell up as it accepts more and more water. It takes in more and more water, and it can potentially burst if the osmotic pressure is enough to overwhelm the cell membrane. So with a hypotonic, the cell swells. The cell swells. So for osmosis, we need to understand osmotic pressure and that water always moves towards a region of higher osmotic pressure. We need to understand if we increase the number of solutes, that's going to increase the osmotic pressure. We will also want to know what happens to a cell that we put in an isotonic solution. What happens if we put a cell in a hypertonic solution? What happens to a cell if we put it in a hypotonic solution? All right, active transport. So with active transport, this is our first active process that we've discussed. And with this one, we have to remember this one costs ATP to do. This is not just some simple passive process that we've been doing so far. This one actually costs ATP to make this happen. So with active transport, we move things from lower concentrations to higher concentrations. So now we're doing the exact opposite of what we've been doing. We're going from lower concentrations to higher concentrations. And what that allows us to do is kind of look for or stockpile some certain hard to get substances. So if we're going from lower to higher, that means we are moving things against the concentration gradient. We are going against that concentration gradient, moving things in the opposite direction. So the way active transport works, it uses carrier proteins, and this is actually what costs us ATP. We often call these pumps. So with our passive transport, we had facilitated diffusion that doesn't cost us any ATP and also uses proteins. With our active processes, active transport specifically uses carrier proteins and costs ATP, and this is why. So what we've got our cell membrane here, but this time I've grabbed a carrier protein instead of a transport protein. So remember last time it was like a little pathway or a little hallway that things were able to pass right through. With our active transport, the solute, whatever it is we're looking for, whatever's going to bind to this receptor, literally gets carried to the other side. So it's going to come down here. Our little solute's going to come down here. It's going to bind to our protein, and then that protein is going to change shape. And as it changes shape, it moves our solute through the body of the protein and pops it out the other side. So our protein physically has to change shape to carry this through, and that's what costs us ATP. It costs us ATP for the protein to change shape. That's something that's kind of going against the grain here. But that is active transport. Couple key things you got to know about active transport. One, it goes against the concentration gradient. And two, we use carrier proteins to carry things across. We often call these pumps, and it costs us ATP to do active transport. Those are all the big things you got to know for active transport specifically. All right, our next type of active transport, or our next active process, I should say, is endocytosis. So endocytosis is where we are making a vesicle, essentially. We talked about this a little bit when we did vesicles. Here we are again. The whole process is called endocytosis. So the cell engulfs the substance in a vesicle. If the substance is a solid, we call it phagocytosis. So this, we think that the cell is eating. The cell is eating. If it's a liquid substance, then we call it pinocytosis. So this is the cell is drinking. And then lastly, we have receptor-mediated endocytosis, which we'll get into in just a second. But for now, let's look at phagocytosis and pinocytosis, because these guys engulf non-specifically. If the cell bumps into it, it's going to try to put it in a vesicle. So the cell reaches out, whatever's there, surrounds the structure, and pulls the whole thing in a vesicle. And that way it's able to maintain the integrity of the cell membrane. So we've got that going on. I've got a generic cell here happening, doing happy little cell things. And it's bumped into this chicken leg. And we know that the chicken leg is the sign of a vesicle, so let's make one. So what ends up happening, cell membrane bumps into that, and it's like, oh, snack. That looks good. I'm going to eat that. And it reaches out with its cell membrane without giving up any integrity. That way we're not letting anything else into the cell membrane, and it continues that process of reaching around until it gets all the way around. Bam. Bam. And once it gets all the way around, it pinches off. So now I've still got the integrity of my cell membrane, but I also now have this delicious looking chicken leg inside of a vesicle. This is a solid, so this is phagocytosis. The cell is eating with that one. So if we were to just switch it from a chicken leg to some molecules of water, H2O, now it's pinocytosis. The cell is drinking. So that's phagocytosis and pinocytosis. Again, they engulf non-specifically. They bump into it, they're going to try to eat it. Now, our third type of endocytosis is receptor-mediated endocytosis. And so we'll do that one up here at the top, where we poke out these little receptors, we'll say, outside of our cell membrane. We just kind of pick them out there. And I always think of receptor-mediated endocytosis as like fishing. We kind of set the fishing line and we can walk away from it, come back and check it later on. It's like fishing. So we've got our little receptors out here, and they're looking for something that is kind of triangular in shape from all the little structures we've got going, floating around out here in our extracellular matrix. We'll do some of that. We'll do some of that. And then let's throw some squares in there as well. So the only thing that's really going to fit is going to be these triangles. So when one of these triangles comes down and lands in this receptor, boom, boom, that's going to trigger the whole thing to sink down into the cell. And kind of in the same way that where we did it with our chicken leg, the cell walls are going to kind of reach up around it, boom, and pulls the whole thing in as a vesicle, receptors and all. And then the whole thing gets sent over to a lysosome to get broken down and see, let's stockpile this stuff. So with receptor-mediated endocytosis, there are really two big key things you got to know. We use the term ligands with receptor-mediated. And ligands are just whatever it is we're looking for with our receptors at that particular moment. So it could be green triangles. It could be a water molecule. It could be a hydrogen atom. It could be whatever it is, sodium chloride or just sodium or just chloride. Whatever it is we're looking for, that's a ligand. And we only, in this class, we only use the term ligands with receptor-mediated endocytosis. So that should be a trigger point for you. Also, the biggest thing of receptor-mediated endocytosis is it provides specificity. It allows us to look for hard-to-get, very specific things that we can stockpile. So now we've got this. We can just hoard it. We can just hold on to those until we need it, especially if it's something that's hard to get. If it's hard to come by, it's much easier to set these little fishing lines, set these little traps out, and wait for it to come to us while we still proactively work on kind of everything else that we need. So that is receptor-mediated endocytosis. So the last two really fall under that phagocytosis umbrella as well. We have exocytosis, which is just the reverse of endocytosis. So with endo, we bumped into that chicken leg, we reached out, grabbed it, pulled the whole thing in. With exocytosis, we're doing the exact opposite. So I've got a cell here. I've drawn in my Golgi apparatus, and this Golgi apparatus has just finished refining this protein I've drawn in red. So the protein is red. Now it's in a vesicle. That vesicle is going to fuse with the cell membrane, and then it's going to open up, boom, and spill out its protein. Wrong color red, but you get the idea. So the protein then moves out, boom. That is exocytosis. We're pushing something out of the cell without giving up the integrity of our membrane. Transcytosis is simply endocytosis that's quickly followed by exocytosis, and this allows substances to transverse or cross the cell. We're going to pop in one side of the cell, and we're going to pop out the other. It's just a thoroughfare for us. And so we'll have that over here where I'll just do this red protein again, pop it in on one side, boom, 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 boom. It's going to fuse with the cell membrane, get opened up, and then it's going to move quickly through the cell and pop out the other side kind of in that same process. That is transcytosis. It allows us to transverse the cell. This is how HIV is able to cross the epithelial layer to move through a cell. So as it moves through the cell, I should probably point out it's still in a vesicle. I should have kind of done it like that. And then it fuses at the other side to get across. So that is cellular transport, both our active and our passive processes. Make sure we're familiar with those by sight and description. Good luck.